All right, so thank you for the introduction. I'll write less next time. So this is really going to be me proselytizing for high-resolution typing and solid organ transplant and thinking about that in virtual cross-match. But I wanted to frame it in a different way this time, thinking about the barrier that people frequently mention that, oh, my lab is small, we're only a solid organ program, we can't really bring in NGS. So start with the standard disclosures that one lambda isn't paying me or telling me what I can or can't say. And so why NGS? So you guys, everyone will see this slide, some version of this slide a million times this week, that HLA genes are the most diverse genes in the human genome. There are over 30,000 identified alleles. The majority of the sequence diversity is actually potentially functionally relevant for stimulating the immune system. There are over 18,000 protein coding variants. And this huge number of genetic variation means that when we have we need a technology for genotyping that's going to let us distinguish the wide array of possible alleles and do so in a robust method where we're confident in the answer that we get and is going to eliminate or minimize ambiguity. So again, this amount of variation is actually pretty important. We think about it, if we think about sort of the structure of HLA, you can see a crystal structure, a cartoon of a crystal structure on the far left side there. So that's A2, and I've highlighted sort of the two areas that are important in thinking about stimulating the immune, how we traditionally think about genotypic variation, which is that there's variation in amino acids that are on the outside of the molecule, highlighted with the red circles. Those are the amino acids that dictate antibody reactivity. We typically think of that as the allele group or sort of first field typing. And then there's variation that occurs within, feedback, there's variation that occurs within the peptide binding groove highlighted by the black bars. That affects how T cells are capable of seeing tissues as self versus foreign. And we typically think about that variation being encoded in the second field or high resolution genotype. And just thinking again about why it's important, why do we have to care, why is it so important to be so accurate? I love showing data from two studies here, one of which, and bone marrow transplant really tells us, right? So in the 1960s, we started out with serologic HLA typing. We could only identify a limited number of polymorphisms. In the 1980s, we began to move toward a molecular analysis, but it was low throughput. We could only target certain areas of certain genes. And using that type of technology, we were able to promote and begin doing matched unrelated donor transplants. But throughout the 80s and 90s and even the early 2000s, we had the problem that's illustrated on the survival curve on the left-hand side, where we're looking at treatment-related mortality, and we were comparing HLA identical siblings in the black line at the bottom with the low level of treatment-related mortality incidence. And then we have unrelated mud donors. We can think about traditional genotyping with the dotted line on the top showing a near 50% incidence in treatment-related mortality by year 1. Well, what was discovered in the 2000s was that we were not adequately resolving HLA genotypes, and we maybe weren't, there was room for improvement on matching donors and recipients for bone marrow transplant. And as sequencing came into, DNA sequence analysis came into the clinical labs and clinical application, we were able to identify that the issue for stimulating the immune system wasn't necessarily that it was simply an unrelated donor, but it was really the degree of mismatch that was driving the outcome. And again, by technological advancement and how we were able to provide HLA genotypes, we were able to change clinical practice and really facilitate mud transplants in BMT. So getting to next generation DNA sequencing, so NGS simply refers to DNA sequencing methods that generate DNA sequence in massively parallel approach. There are two sort of main applications within the clinical space. You can think about Illumina or ion torrent platforms, both of which use different chemistry approaches, but basically just you're reading the DNA as the sequence is being synthesized in the instrument. So NGS approaches enable high throughput sequencing of large amounts of DNA. So the total sequencing capability is actually really relevant to HLA because HLA are relatively large genes. We are interested in 11 genes. There's a lot of genetic variation. So we really need to generate a lot of sequence. Another benefit of NGS is that it's stranded. It provides information on the cis and translocation of variable nucleotides. So most common situation is that for every HLA gene that we're interested in, there are two different alleles, a different allele on each chromosomal copy. So knowing the cis and trans relationship between the nucleotide sequences is very important. So back to what's the lesson that I want to talk about is thinking about entering the NGS world from the scope of being a solid organ transplant. So for the last couple of years, I've been helping out the UC Irvine laboratory, which has historically provided service for a kidney transplant program, transfusion support, and disease association testing. In 2019, there were plans that UC Irvine wanted to begin a stem cell transplant program that was planned to begin in 2020. So we had to think about adding high-resolution HLA genotyping to our lab capacity. So we had to think about the things that we always think about when adding testing, right? Method, instrumentation, workload, workflow, reporting, and cost. And we had to think about this in the framework of having a small lab that really didn't have experience with supporting high-resolution typing, DNA sequencing, or bone marrow transplant. Well, thankfully, I didn't have to build this entire approach from scratch. We have a pretty extensive experience from the UC San Diego lab at this time. So we had begun developmental work on NGS for HLA in 2014. By 2016, we had implemented NGS for high-res typing. By the time we had gotten to planning this in 2019 at Irvine, we had done over 3,000 high-res typings at UC San Diego. In 2018, we'd actually moved all of our solid organ work to NGS, including retrospective typing of deceased donors. So that added another couple thousand cases of experience. So we came into the Irvine with a pretty good head of steam. And we sort of, the benefits of NGS, the sort of analytical benefits of NGS have been described ad nauseum. Just a couple of points. Increased gene coverage by HLA-NGS provides more accurate genotyping. So if we think about what is normally covered even by normal high-res DNA sequencing or by low-res, we're looking at the areas on the gene maps that are highlighted in red versus the areas in yellow, which are what we were able to look at with NGS. That translates to a higher frequency of unambiguous genotypes, as illustrated by the bar graph on the sort of upper left side. And really, this level of ambiguity, this level of decreasing ambiguity helped us with, we know that we're providing a better clinical result, but then there's the issue of practicality. So again, back to the standard, our lab is small, how are we going to make this work? Well, we had learned the lesson at UCSD that integrating high-res HSCT typing in our low-res SOT workflow actually improved lab operation and cost. So the graph on the top shows the average turnaround time for high-resolution typing. So the area in blue is historical using SBT. If we look at the area in red, that's when we were doing NGS for only high-res typing. And then in green is when we added solid organ transplant to our workflow. And what we saw is by adding SOT, we were able to add to our volume. We decreased our time between batches, and we were able to keep our turnaround time where we wanted to be at around 5 days. We usually average about 5 days. Because we increased the number of batches we did in a week, we actually decreased the number of outliers that we had also. If a sample did have a failure, if we needed to go back and do additional testing, we were doing all of that in a more timely fashion, so we were having fewer samples sort of miss our 7-day turnaround time goal. If we think about cost, NGS does have the impractical aspect that there's a high fixed cost per run. There's also the fact that you typically have to buy a new box, and the new box can be expensive. But what we saw at UCSD, at least initially, was that there was cost savings from integrating high-res and solid organ transplant work. So without giving dollar amounts, what we saw is the bar graph on the top right, is that when we were doing, there was some cost X that for high-res typing using only our high-res HSCT workload. But when we increased our workload by adding in SSO, or by adding in the SOT, we actually dropped the total per sample cost to below that of SSO. So NGS became a highly cost-effective approach for us, because NGS is, one of the benefits of the platform is that it's extremely scalable. The other thing, of course, is infrastructure. You know, we had initially started out with MySeqs in the UCSD lab, and for a variety of reasons, we fell out of love with them. But we discovered a new love, which was the Illumina iSeq, which is extremely cost-effective approach to do this. Anyone can ask me after this, I'll tell you why I love that instrument so much. But basically, it's cheap, it's small, and it does exactly what we need to do in sort of a production-oriented way. So we sort of overcame, we had a way to overcome the cost. So then we began to think about method. So we had a lot of experience at the UCSD the UCSD lab with looking at different methods. By this time, we had moved to hybrid capture, but we had experience with the long-range PCR methods from multiple vendors. So thinking about either long-range PCR versus target capture. So long-range PCR can be used to amplify the target regions, the HLA, and then you simply prep your libraries from those amplicons. And it's very similar to workflows that are already in the lab. So the lab is technically familiar with doing PCR, but there are some issues with long-range PCR. Samples with degraded DNA can be problematic. Long-range PCR can generate crossover artifact sequences, and non-amplification of certain target alleles can be a significant issue. So a newer technology is target capture, which uses oligonucleotide probes to sort of pull target sequences out of solution from genomic DNA, and then you do your library prep with minimal PCR amplification. So that's great. It reduces a lot of the negative technical errors that occur in long-range PCR, but it's much more technically challenging. And thinking about the Irvine lab with a small number of technicians with no high-resolution typing expertise, I decided that the best way to approach this was keeping it generally familiar with PCR and within a world that was more comfortable. So we ended up going with the long-range PCR method. The other major factor in this analysis was thinking about data analysis. So HLA sequencing analysis has extra challenges. Again, we have a large degree of genetic heterogeneity. So NGS software has to be able to resolve that. It has to be able to phase the nucleotide sequences. It needs to demonstrate, it needs to do a very good job of telling you, should you be confident in the DNA sequence information you have, or do you need to do additional testing? We were actually really happy with the TSV software. It seemed very intuitive. It provided a clear allele and serologic equivalent assignments. It was the combination of the method being generally familiar and the software analysis being something that felt more intuitive was really a driver for us at the Irvine lab. So doing validation, we began with a parallel testing of 56 high-res samples. 99.9% of alleles tested were concordant with the reference type. There was one discrepancy, but it was in a G group, so it was deemed to be a not clinically relevant discrepancy. There were 4 alleles that required additional testing with SSP, but these were easily resolved ambiguities. In general, this performed well across a range of DNA inputs. I did want to highlight one thing, thinking about as we thought about the quality metrics of the libraries going on. Because we use the iSeq, the cluster density is not a very good metric for us. The iSeq has a pattern flow cell, so it's not like a standard MySeq or the other high throughput Illumina sequencers. A high cluster density is actually it's not possible. So that really ended up not being a good metric for us. But clusters passing filter was a much more important metric, and then of course Q30. So over the first year and a half or so of typing at UCI, we've tested almost 1,000 samples. We have had about 6% of our samples that require additional testing, so they're flagged as requiring additional testing because there's suspected dropout, there's extensive homozygosity, there's ambiguity that needs to be resolved beyond a G or P group, which 6% isn't bad, and we simply resolve most of those with SSP. It's fast, it's easy, and it gives us the answer we need. A couple more extensive ones we have sent out to another lab. I won't say where, but just to see if we got the same answer by NGS. We keep our turnaround time at about 7 days. We're typically batching 1 to 2 runs a week on the iSeq. So again, we're doing batches of. One of the benefits of the iSeq is it has a relatively small cartridge, so we're typically running less than 24 samples. So we would rather run more frequently but fewer samples to delay turnaround time. So in December 2021, we actually migrated to FastPlex. There's the reduced hands-on time from 3 hours to 1.5 hours, although we don't really see much of a difference for us really in the overall turnaround time or the technician sort of workload. We're still at about the same. Really, TAT is driven by batch effects for us. We did a validation study. Again, the validation study performed extremely well. We've tested about 400 samples using this approach. We're still seeing that about 5% of samples are requiring additional testing, and again, the most common problem is still dropout. So going to the next part and thinking about, so how does this fit in for a lab that's primarily solid organ based? So we all know virtual cross-matching is useful in facilitating organ allocation. It's becoming not just an option, but a necessity as we've gone to sort of a broader regional allocation. We have national allocation for a lot of our patients. And if you look in the literature, virtual cross-match is great. It has 90 to 95% accuracy in predicting cellular cross-match results, but that's partially true, right? So I highlight 2 studies that we've done. One that I did a while back using very controlled dilutions of sera, and with identified DSA, we used controlled dilutions of serum and titered cross matches, and very tightly understood how much antibody was present. And the ROC curves on that left figure are fantastic, right? Those are like 97, 98% accuracy. But then the figure on the right is in practice looking at about 1,600 cross matches out of UCSD for deceased donor organ allocation. And the area under the curve there is still pretty good, but it doesn't look like the figure on the left. So what's the difference in the theory versus reality of virtual cross-matching? Well, there are sources of error, right? We know that the SAB assay isn't perfect for a whole bunch of reasons I would be happy to talk about at some other time. There are lots of false negative, false positive interactions. MFI is not a great indicator of antibody strength. But the other issue is that there's incomplete or inaccurate donor HLA typing. So what we typically see when we're evaluating a deceased donor, trying to understand what the HLA typing is the output, something like the output on the left, where it's not important that you read it, it's important to understand that there are a great number of potential alleles present with any of the low-resolution testing methods. And the low-resolution testing methods are very, very good at determining low-resolution genotypes. So if you want to predict reactivity against the 120 WHO antigens and maybe another 50 or so well-characterized serologic splits off of that, the low-resolution typing methods are really good at that. However, it's not very good at estimating high-resolution genotypes. So on the right, we have a review of 296 deceased donor typings performed by standard RT-PCR. So on average, at every locus, there's between 3 and 5 common well-documented alleles that are suggested as potential genotypes within the same serologic group. And when we review, we actually see that in a small number of cases, there are the actual allele confirmed by next-generation high-resolution typing is not included in the allele string at all. So it's important to remember that low resolution is giving you an idea, but it's not giving you the right answer, and it may not be, the right answer may not even be included anywhere in that allele string. So how does that variability, that ambiguity impact virtual cross-matching? Well, again, we think about virtual cross-matching in serologic antigens with thinking about those 120 WHO antigens, thinking about, oh, I know the splits of 205 or whatever. But there are a lot of, there's antigenic variability within serologic within serologic groups that's actually at the allele level. So we have a table on the left here just showing amino acid variation for alleles within A2. Superimposing where the locations of those changed amino acids are back on our cartoon structure, we can see that some of those are on outward-facing residues on the alpha helices, areas that we know interact with antibodies. So there are definitely allele-specific antibodies that can cause immunologic incompatibility, and that complicates the virtual cross-match. So doing a review of about 2,500 cross-matches at UCSD, where we have donor allele level typing confirmed by high-res NGS, we saw that there were about 3% of cases where having allele-specific potential DSA was essential in evaluating compatibility. In general, we tend to think about 7% of our patients have antibodies that fit this sort of allele-specific pattern. The literature on this is sparse. Our estimate is at the low end. The other couple of papers that are in the literature suggest maybe closer to 15%. I think it's a matter of how you evaluate allele-specific specificity, but for us, these are clear examples where at the top you can see there's a difference between, say, 3301 and 3303, and on down, and based upon the specific allele that's present, that is what correlates with the reactivity in the physical flow cross-match. Again, at our center, so we had 15% of samples with allele-specific antibodies for this. This is actually, that's a bit of an overestimation for us. There's some, again, the math, I would say the math is debatable, whether it's 7%, 15%. There's another higher estimate in the literature. But again, it's not uncommon. We're in the general range of 10% of patients have this as a potential complicating factor when trying to assess their compatibility with an organ for cross-match. The value of knowing that information probably varies by location. So HLA allele frequency relates to ethnicity. For us in San Diego, we tend to see some donor genotypes, some high-resolution genotypes that are less common in other parts of the country, B1517, DR407, DR411. So it's really helpful for us to be able to evaluate that high-resolution genotype for some of these patients. And again, as donor sharing is increasing, we all sort of need to have an increased awareness for unusual antibodies. So evaluating that, sometimes you have to go to the sequence alignments, look at the predicted protein sequence, and try to understand what the reactivity is going to look like. Here's an example looking at DR407. You can see there's a replacement of the lysine at position 71 compared to 401, but that same amino acid position is shared with DR404. If you look at the antibody pattern, you can see that there's a pattern where 404 is specifically reactive, and then the patient is reactive against DR407. So sometimes this requires a little more interpretation from our end on the virtual cross-match. But in general, being able to have high-resolution genotyping for donors is extremely helpful. And it's feasible for even solid organ-only programs to really bring that technique in and work with this. So I just want to thank everybody, both at UCI who helped bring this in, and then at UC San Diego who sort of helped provide the foundation for the work that we did a little further north. Thanks.